Alrighty, hi guys, and welcome to my video, Solving Problems in Mendelian Inheritance, Part 3, Basic Variations. So parts 1 and 2 were for week 6. So this is part 3, which parts 3 and 4 are going to be for week 7 here. And the goal here is to help explain some of the new modes of inheritance that we picked up this week, and how they work and how to go about solving problems like that. So we'll take on 4 of the more basic modes of inheritance in this video, and then I'll tackle the more tricky ones, the more complex ones in the next video. So in this video, we're going to cover codominant inheritance, also known as codominance, incomplete dominant inheritance, also known as incomplete dominance, sex-limited inheritance, and then we'll finish out with lethal alleles. So each of these, the nice thing about this week, in my opinion, it's going to be easier than week 6, because in week 6, we were trying to hammer down the ideas of how Punnett squares work and how you read the problems and how you extract the information out of them. This week, we're not changing any of that. The only things we're changing, at least for this video, is how you interpret a Punnett square. That's the only thing we're going to do differently here. Everything else from week 6, you know how to do for this week. So we'll just go through these, and then in the next video, we'll hit the more complex stuff. Now notice a bunch of these will be on your homework assignment. Here's your homework assignment here. But I am not going to cover them in the same order they are included on the homework assignment. So just be aware of that. I am covering most of the things that you will find on here. Questions 4 and 5 are going to be in my second video, but 3, 2, and 1 will be in this video. Okay. So let's start with codominant inheritance. Codominant inheritance is, you actually encountered this accidentally in week 6 when we were doing the calico cat problem that frustrates the entire country. It's the worst because we didn't teach you codominance yet. So now we get to go in and explain this. Codominance, what the definition of codominance is if you're a heterozygote, meaning you have a heterozygous genotype. You got a big R, little r. Let's come over here. Speed this up a bit here. Okay, so what codominance says, inheritance, and my handwriting is struggling today. Okay, what codominant inheritance means is if I'm a big R, little r, I'm not going to be what you think. What we learned in week 6 is that, you have a dominant allele and a recessive allele, and you can use whatever letter you'd like, right? So let's pretend we're in flowers. This is a very common example for codominant inheritance. Let's pretend we're in flowers, and the big R allele is red, flower color, and little r allele is white, flower color. Okay, we know this. This is standard basic stuff from week 6. Well, if we were to work out our little charts like we did last week, if I'm big R, we would expect them to be red in color, right? Because the big R allele, that makes them red. We established that earlier. According to week 6, if I'm big R, little r, I'm going to be red because the big R will overpower the little r and make me red. And then if I'm little r, I'm a white flower. But codominant inheritance is going to kind of change up how this works. Instead of being big R, little r, because like I said at the beginning there, codominant has something to do with altering what big R, little r means. If I'm big R, little r in a codominant inheritance situation, it won't be a red flower. Instead, Oh, dang it. I knew my eraser was going to be too big. Okay. Red, white. Instead, I'm going to be a red and white spotted flower. So basically you would wind up with a little flower that would look like this kind of thing. We got our little tulip or whatnot. But rather than being all red, it would be spotted like this. We saw this with the calico cat problem last week, where you expected an X big B, X little b female to be yellow because the big B was the dominant allele and the little b was the black allele. But because she was heterozygous, X big B, X little b, she was calico with both black and red on her body. That's a codominant situation. Anywhere where you're going to find spots, that's codominance. And I'll show a different or another example in my second video where we run into codominance as well. But the classic example for codominant is if it makes spots, it's codominant. Basically, the reasoning behind this is neither allele. We say that we say one of them is dominant and the other one is recessive, but in reality, they are codominant to each other, meaning this is kind of an arbitrary designation. So we just decided that the big R was going to mean red and the little r was going to mean white, because in a codominant situation, neither the big R or the little r are strong enough to overpower the other one. So they both show up in like spots, in patches. They each have their own little territory that they show up in. So you will have a flower that has red spots and white spots. Sometimes it looks like it's like splattered paint on them and stuff like that. You get some really cool patterns out of these flowers, and floral designers love these kinds of things. But that's a codominant problem. The only difference between a codominant problem and the ones we did last week, which are known as complete dominance, where one allele totally overpowers the other one, the only difference between that and codominance is what a big R little r means. Instead of being whatever the dominant allele is, the phenotype is spots of the big one and the little one. So yeah, that's codominant inheritance right there. You get spots. Again, if we do a Punnett square here, okay, and I've got my big R is equal to the red allele, and the little r is equal to white. Let's say I cross a red parent with a white parent. When we say cross, again, that just a reminder, that means they're just married to each other and they're having fun together. So if we draw out a Punnett square, it's going to look exactly like we did before. You would have, in order to be red, you'd have to be big R, because remember, in this case, last week we had to clarify whether they were big R or little r, or big R or big R little r, because the heterozygous phenotype looked exactly like the dominant phenotype. So you had to go look at the parents and all sorts of things like that. The really nice thing about codominance is it's obvious whether they are heterozygous or homozygous without having to look at the parents, because a heterozygous phenotype looks different from a homozygous one. So it's great. It's even easier than last week. Isn't that wonderful? So in order to be red, they have to be big R, because if they were big R, little r, they would be red and white spotted. And then for them to be white, they have to be little r, because if they were big R, little r, they would be red and white spotted. So we know one parent is big R, the other parent is little r. So we've got a big R up here and a little r right here. So you would wind up with, in this case, my little example here, all of them would be big R, little r. So your phenotypic ratio would be 100% chance of red and white spotted. Notice it's not going to be 100% red because we're in a codominant problem, not a complete dominant problem. So all four of those squares will make a red and white spotted flower. If we were to put two red and white spotted flowers together, so now I'm marrying, so we'll change this up here. Okay. Okay, so now instead of marrying a red and a white, let's marry two spotteds. Spotted crossed with a spotted. So in order to be spotted, they have to be big R, little r, because that's the only way you can be spotted. Because again, this is a codominant problem. So a big R, little r will be spotted, red and white. So this one would also be big R, little r. So you run your standard Punnett square, again, just like we did last week. You come out with big R, big R, big R, little r, big R, little r, and little r, little r. Now, because there are three possible phenotypes here, you actually have to go out and state the phenotypic ratios of all three colors here. Because I have all three colors present, right? This one up here, that's going to be red. This one down here, that's going to be white. And this one down here, this is going to be a red and white spotted flower. So so then I would write out my phenotypic ratio as 25% red, 50% spotted, red and white spotted. I would write that out, but for sake of time, I'm not going to. And 25% white. Because that one makes a red flower, that one makes a white flower, and those two make spotted. So, and remember, this isn't saying that if they had 4 kids, one of them will be red, 2 of them will be spotted, and one of them will be white. Remember, a Punnett square says the odds of having a kid of each color from each pregnancy. So they could have all white kids or all spotted kids or all red kids. It's all just a flip of the coin for each pregnancy. Okay, so that's codominant inheritance. We beat that one to pieces here. So really the only difference here is just what big R, little r means. It's not red, it's in fact spotted. Okay, let's work through an example problem that isn't working with colors, because virtually every codominant one you run into in your homework and stuff is dealing with colors. So let's work with one that doesn't deal with colors, but is still a codominant problem. This one is a color one. I forgot. We'll use a different problem later then. Okay, in chickens, red feather color is codominant to white feather color. Heterozygous individuals are spotted red and white. So there you go. Notice it says in chickens, red feather color is codominant to white feather color, and heterozygous individuals are spotted red and white. Even if I told you that, even if I didn't tell you that red is codominant to white, reading this statement, heterozygous individuals are spotted red and white, gives it away. That's codominant. If it spots, it's codominant, guaranteed. So you don't even need me to tell you it's codominant to know that it's codominant, because heterozygous individuals are spotted red and white. That's a codominant problem. A red rooster mates with a white hen. What is the phenotypic ratio of their offspring? Okay, so we'd come in here. We have a red rooster. So the first things we do, remember there's 3 steps to solving one of these things. We go in and we determine what is normally we would determine what is the dominant allele and what is the recessive allele. In this case, neither one is dominant or recessive. So we're just going to deem one of them to be the big R and one of them to be the little r. So we've got the red allele and the white feather color, the white allele. Okay. Now the second thing we have to do is write out all 3 of our possible genotypes here. Got a big R, little r, and little r, little r. If I'm big R, I'm red. If I'm big R, little r, I'm not red. I am red and white spotted, because that's what the question told me. And if I'm little r, I'm white. So if you have a red rooster he's going to be big R, big R. And if you have a white hen, it'd be little r, little r. You have big R, little r, big R, little r, big R, little r, and big R, little r in this case. So 100% of their offspring, or 100% chance of a red, white, spotted offspring child. And there you go. That's how a codominant problem works. Okay. So now that we've covered codominance, let's go to the next one in line. The next one on our list is incomplete dominance. Now, incomplete dominance is very easy to get mixed up with codominance because they behave similarly but have different end results. With an incomplete dominance question, incomplete dominance. Again, the only difference between this and complete dominance that we worked with last week is going to be what a big R, little r, what a heterozygous means. It's exactly the same as we did with codominant. The difference here, though, is a big R, little r, rather than being spotted, having both of the original phenotypes show up at the same time, neither allele is actually strong enough to fully overpower the other one or even exist by itself if it has to fight. So instead, they blend together. The classic example, again with flowers here, is for incomplete dominance, is if we have, again, we will say big R is equal to red flower color, little r is going to equal white flower color. But instead of big R, so let's write these out. We've got big R, big R, big R, little r, little r, little r. Big R is still going to be red because we've just got red alleles. Little r will still be white. But instead of big R, little r being red like it is in codominant or in complete dominance, or red and white spots like it is in codominance, it's going to be pink. Notice pink wasn't one of the original options. You were either red or white. But we are now pink because in incomplete dominance, the alleles blend together to create a phenotype that is somewhere in between the two of them. So you're still one solid color rather than being spots like you were in codominance. But that one solid color is a blend of the other two. So big R means you're red, little r means you're white, big R, little r means you're pink. And other than that, you're going to run your Punnett squares exactly the same. Just note that if you have a pink parent, they're big R, little r. So again, this one will be way easier than it was last week because you don't have to look at the parents to determine if they're big R, or big R, little r, because big R, little r's look different than big R's. It's wonderful. So I just have to tell you that they're a pink parent and you know they're big R, little r. And so then again, you'll report the percentages you'll report the percentages accordingly. So you'll say a certain percentage of their kids are red and a certain percentage of their kids are pink and a certain percentage of their kids are white, etc. So let's work through a practice problem. This is the one that I have for you that is not color. So let's go look at this. In humans, curly hair is inherited in a pattern of incomplete dominance. Individuals homozygous for straight hair will have straight hair. Individuals or homozygous curly individuals will have curly hair, and heterozygous individuals will have wavy hair. Notice this is an incomplete dominance problem. Yes, we're not working with color here, but notice if I have straight hair, right? I've got one allele that is straight hair and one allele that is very curly hair. Then the in-between, if I were to blend these two together, it would create wavy hair, which is something in between the two. So this is not a color question, but it's still incomplete dominance because if you're big R, little r, you have something that's in between what the little r allele says and what the big R allele says. So you get a third phenotype that wasn't one of the possible alleles. So we're still doing an incomplete problem here. Okay. Okay. All right. If a straight-haired man marries a wavy-haired woman, what are the odds their first child will have wavy hair? Okay. So first off, first step to every problem, determine what your two alleles are. Again, dominance and recessiveness doesn't really matter because they're incompletely dominant to each other. So I'll just decide that having straight hair is going to be my big R, and having curly hair is my little r. And then all the three possible combos, we got big R, big R, big R, little r, little r, little r. This gives me straight hair. This gives me wavy hair, because that's what they told me. Heterozygous individuals will have wavy hair. And this gives me curly hair. Okay, so we have a straight-haired man. So he is going, according to our table here, he's going to be big R, big R. It's really helpful writing out that little table. Most people skip that, but it's really helpful, especially this week, to write out that little table, just so that you don't accidentally confuse yourself and run back to week 6's ideas that big R totally dominates little r. Okay, so our big R1 is going to have straight hair. And then we have a wavy-haired woman. So according to our table, big R, little r means wavy. So she is big R, little r. So we're going to have big R, big R, big R, big R, big R, little r, and big R, little r. So our phenotypic ratio here is going to be 50% straight hair, 50% chance of straight hair, child, and a 50% chance these two are going to be wavy hair, so it'd be a 50% chance of having wavy hair. So there you go. That's it. There's an example of incomplete dominant inheritance that isn't involving colors, but the color one is a great example because red and white, you blend them together like paint. That's what inheritance or incomplete dominance is the third phenotype. If you're a heterozygote, it's a blend of the other two, whether that is you're blending red and white to make pink, or you're blending straight and curly to make wavy. Either way, it's a blend, whereas codominant is both alleles are showing up at the same time, independent of each other. So if we were doing this example with hair and it were a codominant problem, I would expect the big R, little r one to have both straight and curly hair. It would be kind of like that would be a really weird hairdo. You'd have like an afro with hair coming off the bottom. That would really mess everything up. So, but there's the difference there. The only difference between incomplete and codominance and complete dominance is what big R, little r means, what being a heterozygote means. Okay, there's incomplete dominance. Let's go look what's next in line. Okay. Sex-limited inheritance. These are where things start to get a little trippier, and where we get even further away from what we did in week 6. With sex-limited inheritance, let me come back here and erase all this. Sex-limited inheritance is the easiest one we've covered so far, but it's also the one that you have to make sure, or you have to overcome the most mental blocks here. So we got sex-limited. Inheritance. Okay, so for sex limited, the trick here, this is not with big R, or this is not a difference with big R and little r. This actually has something to do with how the phenotypes are manifested. With these ones, often you will have a big R allele and a little r allele again, and big R will mean something and little r will mean something. But the one or the other allele, or neither allele, just simply doesn't show up in one gender. It doesn't matter what they have. So for example, a common one, let's say we have, let's go back to my chicken. Let's go to chickens. Actually, let me check to see if that's my example over here. Okay, yeah, we'll just work through this question here because I think it will help. Okay, in chickens, gobblers manifest as an X-linked trait. That's like their little thing underneath the throat. Okay, in chickens, gobblers manifest as an X-linked trait. Gobbler is dominant to no gobbler. Okay, so let's look at this. So first off, we're here in X-linked again. So remember, those first two, we were doing autosomal problems. Now we're on X-linked, so you can't forget our X-linked stuff from last week. So we're on an X-linked trait. So we have to come in here and determine what is our dominant allele and what is our recessive allele. In this case, it's not incomplete in codominance, so we're back to having one allele that's more powerful than the other one. We've got, let's see, in chickens, gobblers manifest as an X-linked trait. Gobbler is dominant to no gobbler. So big R means gobbler, and little r means no gobbler. Okay, females never have gobblers. We'll come back to that. What is the resulting phenotypic ratio of the offspring from crossing a gobbler male with a heterozygous female? Okay, so a gobbler male, he's got, notice this is, now notice, at first you might think, uh-oh, is he big R or is he big R little r? Well, remember, this is an X-linked problem. So if we write out our table, you actually have 5 possible combos you can be, right? You could be an X big R, X big R. You could be an X big R, X little r. Be an X little r, X little r. You could be an X big R, Y, or you could be an X little r, Y. If I'm an X big R, that would, running off of what we've learned last week, and I'll come and amend this later, X big R would be a gobbler female. X big R, X little r would be a gobbler female. And X little r would be a no gobbler female. And then for the men, X big RY would be a gobbler male. And X little ry would be a no gobbler male. Okay, that's how it would behave under week 6 conditions. The problem here, though, is this is a sex-limited inheritance problem. And I never said sex-limited in here anywhere. The giveaway sentence is right here. Females never have gobblers. With sex-limited inheritance, one gender just simply does not express one or either of the two phenotypes, no matter what their genotype is. They just simply don't do it. So these ones, if you read the question, they're actually the easiest thing, because if it asks you for a percentage of females, you can just go and check either 100% or 0%, depending on what it is. So let me show you this example here. So because females never have gobblers, if you have an X big R female, she does not have a gobbler because she's female. And if you have an X big R, X little r female, she does not have a gobbler because she's female. So that's also a no gobbler female. So notice, even though big R is dominant to little r, I told you in the question, females never have gobblers. So so that doesn't mean that females can't get a big R allele. It just means that they will never have a gobbler. So they can have whatever genotype they darn well please, and they will never have a gobbler because they're female. It's a sex-limited trait. So we have the resulting phenotypic ratio crossing from a gobbler male. So we look at our table. The only way to be a gobbler male is to be X big R Y and a heterozygous female. Notice it wouldn't help me to say a non-gobbler female because females never have gobblers. So I had to say heterozygous. So she's X big R, X little r. Then we do that cross. Got an X big R, X big R. X big R Y, X big R, X little r, and X little r Y. Now notice, because this is a sex-limited inheritance problem, and I know females can never have gobblers, I don't even have to look at the Punnett square. I know that 0% of their daughters will have a gobbler. Because the question said females can never have gobblers. So even though I've got an X big R female there, and an X big R, X little r female there, neither of them have gobblers because this is a sex-limited inheritance problem. So the big R doesn't matter because they're female. Then over here, now let's look at the men. X big R Y, that means gobbler. So we've got 50% sons gobbler. And then you could either just leave it at that, or to be more specific, you could say 50% of sons no gobbler, because X little r Y means no gobbler. So notice, daughters never have gobblers. So we just said daughters never have gobblers. Even though both of those under week 6's rules would have gobblers, daughters never have gobblers. So daughters never have gobblers. So just make sure to read in those questions. Doesn't matter what you get out of the Punnett square for a daughter. It's that you just follow what the instruction says for the daughter. Or if it says the sons are missing out, then do it for the sons. Just read the question. And it doesn't necessarily have to be a sex-linked question either. Like you could have an autosomal question, where if you had a big R, little r here, like for flower color, for example, and I'm just borrowing the Punnett square. Ignore this question. We'll come back to it. Okay. If we were to do a flower color one where big R meant red and little r meant white, and I told you, and if I told you in the question that that males, I mean, flowers don't have a gender, but bear with me, that males are always white. Notice this is an example of an autosomal problem that would be sex limited. Then if you had a big R man crossed with a little r woman, then we would have a big R there. I'd have a little r here. You would wind up with this. And remember, all 4 squares apply to both genders in an autosomal one. So you would look and pretend all 4 squares are sons. Well, it says males are always white. So all 4 of them are white. So it'd be 100% white sons. And then for the daughters, all 4 squares are daughters. You would have all of them would be red because the big R overpowers the little r. So you would have all red daughters. So there's an example of an autosomal, just a brief, quick example of an autosomal sex-limited one. And here's a sex-linked sex-limited one. But in either case, one of the 2 genders just simply cannot express one of the phenotypes or either of the phenotypes. And a great example that I use on my practice exam is beards. Men can have varying qualities of beards, and women just don't have beards. So that's a sex-limited trait. Is men can have multiple ones that have multiple alleles that influence their beards, but women who have the genes for beards just don't turn them on. It's a hormonal thing. You never turn them on. Okay. It turns out most sex-limited traits are secondary sexual characteristics. So like beards for men is a sex-limited trait. Breast development for women is a sex-limited trait, things like that. Where men have the genes to develop breasts and women have the genes to develop beards, but in most cases, they don't turn them on because it's a sex-limited trait. Okay, so there's sex-limited. Now we're on the last one, lethal alleles. This one's the trippiest of all the 4 I'm covering in this video. That's why I saved it for last. Sorry, I talked too much. Okay, here we go. So lethal alleles. Lethal alleles are fun, not for the person getting them, but for the Punnett squares and the phenotypic ratios, because lethal alleles really mess with your phenotypic ratios. You can have lethal dominant alleles, or you can have lethal recessive alleles. The one on your homework is a lethal dominant allele. I'll show you an example of a lethal recessive and a lethal dominant allele. Let me see which one I did over here so that I know which other one to do. Okay, so we'll do a recessive one over here. Okay, so over here, let's say, often what happens with these ones is they are involved with genes that are kind of essential for functioning. So there's an example, for example, there's an enzyme called acetylcholinesterase. Say that 10 times fast. We abbreviate it to ACHE, acetylcholinesterase, and its job is to make your nerves fire or stop firing when they're supposed to stop firing. So your nerve fires a signal to the muscle to contract, and then ACHE comes in and turns it off so that you're not constantly contracting your muscle. Well, if you get a mutation in the ACHE gene that makes the ACHE protein not work, right, a missense mutation or a nonsense mutation, there's your week 5, start panicking, then that would be really bad, because if the ACHE enzyme doesn't do its job, then anytime you send a message to your muscle, you can't uncontract your muscle. In fact, if you've ever heard of sarin nerve gas, this is how it works, is the sarin nerve gas takes out ACHE, which means all your muscles contract at maximum strength permanently, and you suffocate to death because you can't unlock your muscles to breathe, which is a really bad way to go. And that's why using nerve agents worldwide is illegal. Anyway, so following my example here, having a big R allele is being normal. So here's normal. And having a little r allele is being dead, because if you don't have the ACHE protein working, you're dead because you can't breathe, right? So if you are big R, then you're normal. If you are big R, then you're normal. And if you're little r, you're dead. Now, with these ones, with lethal alleles, they never go to full term. So if you have a baby that gets in this case, if you have a baby that gets 2 little r alleles, the embryo will miscarry. You will have a miscarriage before the baby is born. So let's see what this does to our Punnett squares. If I were to do a Punnett square, let's say I've got 2 heterozygous parents. Actually, let's start with a big R parent marries a big R parent. Let's say that. We'll start there. Okay, if I have a big R parent and a big R parent, nope, both of them are normal. If we do our Punnett square here, big R, big R, big R, little r, big R, big R, big R, big R, big R, little r, and big R, little r. All of their kids are normal. Woohoo! Right? So you'd have 100% of their kids would be normal in this particular example. Well, what if we change things up ever so slightly? And this is where things get messed up. Oops. Just shrink down my eraser again here. What if instead of doing a big R cross to a big R, little r, we cross 2 big R, little r's? So over here, I will have a big R, little r, and down here, I've got a little r. Now, the way you would write, the way your brain is going to tell you to write out this phenotypic ratio is to write out, you would have you would have 20, or you would have 75%. You would have 75% normal and 25% dead, right? That's how your brain says to work this out. But with these lethal allele ones, they're going to mess with your phenotypic ratios because being dead is not a phenotype. Being dead is a state of being. So because a kid that has a little r combo is never born, from a scientific perspective, you can never tell they actually ever existed in the first place. Which means we actually don't count the little r square as one of the possible options. They don't count in the phenotypic ratio. So it's as if this bottom square here doesn't exist. So you only have 3 possible options here. So you would actually work in thirds, and I'll show you an example over there. In this case, they would be they would have 100% of their kids would be normal because the one combination that is little r doesn't develop into a kid. So all 3 of their possible kids are normal. Let's work through an example where let's go work through an example that's a little better demonstration of the percentages than this. Let's see. Here we are. Okay, achondroplasia is an autosomal dominant dwarfism disorder in humans caused by a mutation in the FGFR3 gene. Okay, so notice this is autosomal dominant, so we're back on the autosomal. Get my pen out here. Okay, we're back on the autosomal. So I've got, so let's look at my big, oh boy, let's not do that. Okay, we've got to figure out what's my big R and what's my little r. It says achondroplasia is an autosomal dominant dwarfism disorder. You recognize that mode of inheritance from last week. Autosomal means it's on one of the autosomal chromosomes, and dominant says achondroplasia is the dominant allele. So this equals being a dwarf because that's shorter to write than achondroplasia. And so then the little r is being normal height. A heterozygous individual is a dwarf. Individuals who are homozygous for the dwarf allele miscarry before birth and are never found in the population. Okay, so notice this one isn't going to behave like you would normally expect it to behave, because according to the way you would think from week 6, big R would be dwarf, and big R would be dwarf, and little r would be normal, right? But what the question is telling you is if you're big R, you're dwarf, and if you're big R, you're dead. Which kind of seems to break your idea of an autosomal dominant question. In reality, this one's kind of sort of behaving more like an incomplete, isn't it? An incomplete dominant one. But for the sakes of the question here, just treat it, or just write them out. You know, a big R, little r is dwarf. A big R is dead. Okay. So if we have, what are the odds that a child born to 2 dwarfs will also be a dwarf? Okay. So we've got so to be a dwarf, you have to be big R, little r, because if you are big R, you're dead, not a dwarf. So this kid here would be big R. This one here is big R, little r. This one here is big R, little r, and this one here is little r. Okay, so what are the odds that a child will be a dwarf? So we see big R, little r, so that we would normally want to say 50% of their children would be dwarfs. But remember, big R, this child down here would be normal. This child up here would be dead, and those two would be dwarfs. And remember, being dead is not a phenotype. So it's as if, so we're going to treat it as if this square does not exist, which means we're actually working with 2 out of 3 of their kids will be dwarfs. So you will actually not be 50%, you will be 66%, because that's 2/3. Isn't that trippy? So with lethal alleles, be careful. If any of the possible, if all of the possible kids survive, then you'll still be working in quarters because all 4 squares survive. But if any of the 3 squares die, then you will be working in thirds for your phenotypic ratio, not quarters. So you will, so this one, you would have 33% normal kids, normal, and 66% dwarfs, because that 4th option doesn't count. Because it dies, it miscarries before it was ever born, so we don't actually know it ever existed. Okay, so there's an example of a lethal allele problem. So yeah, that's the end of my first video. I encourage you to watch through my second video as well. Again, these, what I've covered here in this video should help you answer questions 1, 2, and 3 on your homework. And I'll go cover the more complicated questions 4 and 5 in my next video. So thanks for watching.